The reactions catalyzed by mammalian nitric oxide synthases, NOS, are central to life. Yet, when they become deregulated, NOS are the source of serious inflammatory pathology, including the leading killers cancer, heart disease, and sepsis. NOS are believed to use L-arginine, tetrahydrobiopterin, nicotinamide adenine dinucleotide phosphate, heme, and oxygen to synthesize the radical paramagnetic gas nitric oxide. Nitric oxide is the universal second messenger, involved in cell signal transduction, gene expression, including epigenetic modifications, control of the cell cycle, apoptosis or growth and differentiation, respiration, neurotransmission, muscle contraction, vasoconstriction, the coagulation cascade, coordination of the immune response and resolution of inflammation, pathogen cytotoxicity, and host antioxidant actions. NOS exist in three principal forms: two constitutive, endothelial and neuronal NOS, eNOS and nNOS, which are calcium calmodulin regulated, targeted to the caveolae, and the largely calcium calmodulin independent inducible NOS, iNOS, active during growth and the immune response, at significantly higher expression levels than constitutive NOS, thus producing much higher amounts of nitric oxide, for which hepatic Kupffer cells, macrophages, and polymorphonuclear neutrophils are a principal source. More recently, a mitochondrial NOS has also been added to the canon. NOS function as homodimers. They require a prosthetic heme to be bound for their assembly and are composed of discrete oxygenase and reductase domains linked by a calmodulin loop. The C-terminal reductase domain uses a chain of flavins, flavin adenine dinucleotide and flavin mononucleotide, to transfer electrons from nicotinamide adenine dinucleotide phosphate, NADPH, to the N-terminal oxygenase domain. The reactions that yield nitric oxide and citrulline from L-arginine via the intermediate N-omega hydroxylated L-arginine are a two-step monoxygenation, currently believed to use 5 electrons. However, after nearly a quarter of a century of research, there is no absolute or final consensus on many of the exact details of NOS catalysis, including the exact role of the cofactor tetrahydrobiopterin. Various proposals for heme-based NOS catalysis have been espoused: a radical-type auto-oxidation mechanism, or involvement of a nucleophilic peroxoferric heme, or of the ferryl oxenoid porphyrin radical species, analogous to that of cytochrome P450 oxygenases. An example of one heme-based catalysis scenario involving first a ferric heme superoxo species, then a heme peroxo species, is illustrated here. For a detailed review of heme-based NOS catalysis competing theories and their problems, please see Santolini et al., a reference in the paper associated to this presentation. The protein-bound ferric heme is assumed to be the catalyst for the dual monoxygenation, largely an assumption from its structural presence, and by analogy with its role in other heme enzymes such as cytochrome P450, and because in studies with purified NOS, no other candidate has been observed. Nevertheless, studies with purified NOS may not reflect what actually takes place in vivo. Another unsolved mystery remains. What precisely makes NOS produce a regulated supply of nitric oxide with predominantly beneficial cell signaling, growth regulatory antioxidant effects in health, and yet malfunction with host detrimental consequences in chronic and acute pathologies? Whilst the current nitric oxide pathology paradigm sees nitric oxide as the problem, this hypothesis proposes that the critical factor behind this paradox is the ratio of nitric oxide to peroxynitrite and related species produced by the NOS. This ratio is high in health and low in pathology, whilst a high nitric oxide peroxynitrite and related species ratio is determined by the bioavailability of cobalamin in one of its principal active forms, ADENOSYLCOBALAMIN, as the primary participant in NOS catalysis. Heme-centered catalysis is the considerably less efficient backup reaction in the absence of ADENOSYLCOBALAMIN, and thus the source of pathology when it becomes dominant. For the absence of ADENOSYLCOBALAMIN from NOS is likely to reflect a general deficiency of cobalamin, functional or otherwise, that will also result in decreased availability of tetrahydrobiopterin, in turn associated with an increase in radical production by NOS. Apart from the fact that the problem of NOS-related pathology remains unsolved, there are several facts and clues suggesting that the principal in vivo NOS reaction may differ from that observed in vitro in purified NOS, and that ADENOSYLCOBALAMIN, not as previously proposed by myself, glutathione-cobalamin, is a reasonable candidate. 1. Structural and kinetic features show NOS differ significantly from cytochrome P450s and from other heme-catalyzed oxygenases. NOS shares some structural features with cobalamin-binding enzymes. 2. The NOS oxygenase domain structure is compatible with accommodation of ADENOSYLCOBALAMIN in a position where its cobalt may be reduced in place of the heme. 3. Electron transfer to the L-arginine, bound as the anti-stereoisomer to the heme, occurs at a distance from the heme, so an intermediary electron carrier is needed. Yet tetrahydrobiopterin, a potential electron donor, is also bound at a distance from the heme and from L-arginine. ADENOSYLCOBALAMIN's adenosyl radical may bridge the gap. 4. Several studies suggest that in vitro, the first product of heme-centered NOS catalysis may not be nitric oxide, but nitroxyl anion or other nitrite species. Furthermore, fast reduction of NOS heme ferric iron nitrosyl to ferrous iron nitrosyl prior to the release of nitric oxide and the slow dissociation of the latter leads in the presence of oxygen to generation of ferric iron plus nitrate, not nitric oxide. And this is the NOS futile cycle. Whereas, in ADENOSYLCOBALAMIN-based NOS catalysis, rapid displacement of nitric oxide from the analogous nitrosylcobalamin in the cobalt-2 state by the adenosyl radical prevents the NOS futile nitrate cycle and excess radical generation. 5. As we shall see, cobalamin is consistently associated with nitric oxide's positive effects in a variety of situations. Moreover, expressed in vivo in ADENOSYLCOBALAMIN-rich environments, neuronal NOS yields significantly more NOS protein, and in the L-arginine to citrulline NOS activity assay is between 4.5 to 10-fold times more productive than in vitro. 6. Unlike hydroxycobalamin and glutathione-cobalamin, ADENOSYLCOBALAMIN does not inhibit the NOS. On the contrary, ADENOSYLCOBALAMIN's characteristic dual role as both a generator of catalytic radicals and as the ultimate radical cage and ultimate radical trap within enzymes results in a dynamic chemistry in known ADENOSYLCOBALAMIN-dependent enzymatic reactions, which may also overcome the spatial, energetic, and thermodynamic challenges of heme-based NOS catalysis. All these points will now be illustrated briefly before we look in detail at the plausible ADENOSYLCOBALAMIN-centered NOS catalysis. NOS and cytochrome P450 enzymes share a few common features apart from a heme presence: a flavin reductase C-terminal domain, substrate positioning over the heme, an enzymatic thiolate heme ligand. However, the differences are more significant. Unlike NOS, cytochrome P450s can exist in monomeric or dimeric form, and their alpha helical architecture confers an extraordinary conformational flexibility, enabling cytochrome P450s to continuously remold the active site to fit a very wide range of disparate ligands. By comparison, the NOS active site is relatively inflexible, being composed of complex beta sheet structures extended in wing formation with peripheral helices. This is distinct also from other heme enzymes. Peroxidases, oxidases, catalases are all largely alpha helical like cytochrome P450. Further, unlike cytochrome P450s, NOS have an unusually open structure, as already mentioned, like a baseball catcher's mitt, with each heme bound in the beta sheet palm of the hand. It is unusual, moreover, to find a heme binding site in a predominantly beta sheet structure. But it is not unusual to find adenosylcobalamin and indeed even methylcobalamin bound in enzymes with a core 5-stranded beta sheet structure. Compare the topology of the NOS core 5-stranded beta sheet heme binding site with that of methylcobalamin in methionine synthase and adenosylcobalamin in methylmalonyl-CoA mutase. The NOS heme is also flipped 180 degrees from its usual conformation in cytochrome P450, facing a large exposed distal cavity that's about 20 angstrom across by around about 11 angstrom deep. 11 angstrom is, coincidentally, comparable to the size of adenosylcobalamin, and its binding site cavity in at least one adenosylcobalamin-dependent enzyme, ethanolamine ammonia lyase. The NOS structure contains a Rosman fold, a characteristic of nucleotide binding proteins. But unusually, this has an alpha-beta fold, yet lacks the conserved water molecule of classical Rosman fold-containing proteins, such as cytochrome P450. A subdomain of the NOS oxygenase domain is made up of a 2-helix bundle, alpha-2-alpha, which forms a hydrophobic core. Adenosylcobalamin characteristically binds within alpha-beta Rosman folds in hydrophobic regions containing alpha-helix bundles alongside 5 central beta strands in class 1 and class 2 cobalamin enzymes, such as ornithine aminomutase, glutamate mutase, lysine aminomutase, methylmalonyl-CoA mutase. This is characteristic also of methylcobalamin in methionine synthase. Thus, the 2 alpha-helix hydrophobic bundles in NOS are a potential location for 2 adenosylcobalamins false nucleotide dimethylbenzoimidazoles, which conceivably move into alignment with the 5 core beta strands in each half dimer once adenosylcobalamin is bound above the structural heme. Moreover, Rosman fold proteins all have cofactors containing adenosine, adenine plus ribose, and all interact with the adenosine in virtually identical fashion, binding the adenosine ribose hydroxyls through hydrogen bonding by an acidic amino acid side chain with conserved aspartic acid glutamic acid residues. As seen here, the NOS active site contains conserved aspartic acid glutamic acid residues, which are asparagine and glutamic acid residues in eNOS, in an optimal position to hydrogen bond the 2 hydroxyls of adenosylcobalamin's adenosine ribose right next to the substrate L-arginine, itself hydrogen bonded to aspartic acid glutamic acid residues. The NOS homodimer contains 4 cavities. Apart from the 2 heme L-arginine tetrahydrobiopterin binding pockets, about 4,000 angstrom cubed each, a large solvent-filled intersubunit cavity around 750 angstrom cubed, surrounding a zinc tetrathiolate centre, capable of accommodating an as yet unidentified ligand, and a smaller cavity at the dimer interface, adjacent to the histidine 436-histidine 437 residue pair. In the substrate pocket, Weinberg et al. Observed that a very large interaction surface around 800 angstrom is available for cobalamin, and their molecular modelling studies, with manual computer-assisted docking analysis, have now confirmed my prior imaginative observation, which predicted that NOS may indeed be capable of cobalamin accommodation in the base-off conformation, that is, with the bond broken between the cobalt and an imidazole nitrogen of cobalamin's lower axial dimethyl benzoimidazole. By coincidence, this is the conformation in which methylcobalamin is usually bound within mammalian methionine synthase, adenosylcobalamin and methylmalonyl-CoA mutase, and in all other adenosylcobalamin-dependent class 1 and class 2 enzymes. Weinberg et al. Found it possible to achieve at best an 80% overlap of the corin with the heme. Thus, the corin macrocycle will be just above the core NOS 5 beta strand section, comparable to its position in known adenosylcobalamin binding enzymes. This is an optimal placement if the ultimate free radical cage and trap adenosylcobalamin plus the enzyme is to act as a cap on the open NOS structure and prevent free radical escape. Moreover, adenosylcobalamin is not always buried in its dependent known enzymes. In some, such as glutamate mutase, ornithine aminomutase, and bacterial ribonucleotide reductase, adenosylcobalamin is initially bound relatively superficially. For example, compare the position of the NOS heme and adenosylcobalamin's position here. In experimental work to date, no change in the NOS open structure has been seen with diverse other ligands. However, Rossmann folds are capable of some flexibility during catalysis. In adenosylcobalamin-dependent lysine aminomutase and in ornithine aminomutase, there are significant active site conformational changes and large-scale domain dynamics, but these are entirely dependent on the presence of both the substrate and, specifically, the adenosylcobalamin ligand. In the structure of human enos, the region just beyond the zinc tetrathiolate, only 2 residues away from the bilateral serine residues that hydrogen bond directly to the hydroxyl side chains of the 2 tetrahydrobiopterins, about 12 angstrom, comprising amino acids 105 to 125 in enos, shows potential for considerable protein flexibility and has been dubbed the flexible arm. One possibility is that the zinc tetrathiolate cavity may be compressed to allow for further expansion of the heme cavity, or it may be an alternative docking space for 2 adenosylcobalamin dimethyl benzoimidazoles that could displace 2 of the zinc tetrathiolate thiol ligands with 2 imidazole nitrogens in a zinc finger motif. The high nitric oxide producing NOS of adenosylcobalamin-rich Streptomyces turgidis scabies has just such a zinc finger motif. Since, in each half dimer, the flexible arm is linked directly to the 2 helix bundle, the latter could combine with the core 5 beta strands to form a hydrophobic sheaf for dimethyl benzoimidazole to thread through to the zinc. This would effectively close the NOS enzyme, impeding heme access to the substrate, with formation of a TIM barrel and release of the adenosyl radical into the active site, as in methylmalonyl-CoA mutase. Finally, adenosylcobalamin's corin, being considerably more reduced than the heme porphyrin ring, gives adenosylcobalamin substantially more conformational freedom. This flexibility may also be relevant to how adenosylcobalamin fits into the NOS in vivo. Compared with cytochrome P450s, the heme-centered NOS have quite a distinct response to oxygen, which participates in the catalytic two-step monoxygenation. According to Raman data in NOS, heme-bound oxygen seems to point away from the substrate L-arginine. This is not inconsistent with the primary binding site for oxygen actually being the potentially better placed cobalt of adenosylcobalamin. The possibility that adenosylcobalamin participates in NOS catalysis would also make sense of the unusually slow ferric enzyme reduction, Kr, of the NOS in adenosylcobalamin's absence, in comparison not just to cytochrome P450, but to other flavor heme enzymes. NNOS, the Kr is 3 to 4. INOS, 0.9 to 1.5 Kr. ENOS, 0.1 Kr. As opposed to cytochrome P450 BM3, 99 Kr at 15 degrees centigrade, or flavor hemoglobin 150 Kr at 37 degrees centigrade. This slow Kr makes it difficult for heme-based NOS to couple oxygen activation and heme reduction to substrate oxidation. NOS are thus much less tightly coupled than cytochrome P450s, and consequently prone to generate excess free radicals. However, in spite of the slow Kr, the NOS ferric heme can be reduced in the absence of substrate L-arginine. Compounding the problem is low tetrahydrobiopterin, which is essential for nitric oxide synthesis, a possible consequence of cobalamin unavailability. Low tetrahydrobiopterin increases NOS reduction of oxygen with increased formation of superoxide and hydrogen peroxide at the expense of nitric oxide, and a loss of the nitric oxide peroxynitrite balance. In addition to the unusually slow Kr, apparent KMO2 values, that is, the concentration of oxygen at which the reaction rate is half of Vmax for the NOS, not only differ between isoforms, but in the case of NNOS 350 and INOS 130, though not ENOS 4, are also considerably higher than those of other monoxygenases, whose heme-based catalysis NOS are thought to emulate. Cytochrome P450 and flavor hemoglobin are more obviously coupled to oxygen, 4 to 10 at 10 degrees centigrade and 60 to 90 at 20 degrees centigrade respectively. Moreover, in the isolated INOS dimer, before L-arginine and tetrahydrobiopterin are bound, the heme is notably in a low spin state, unlike the heme in isolated cytochrome P450. This makes sense if the cobalt of adenosylcobalamin is intended to take precedence over the heme ion as the primary target for reduction. There is also a conflict of opinion as to whether nitric oxide is actually the end product of the NOS. Some have failed to observe nitric oxide except in the presence of superoxide dismutase. Thus, it has been proposed that nitroxyl anion, small amounts of other species from its decomposition such as nitrous oxide and hydroxylamine, and also peroxynitrite from superoxide and nitric oxide, may be the primary NOS products. Evidence shows that the first observed product of NOS catalysis is a ferric heme nitric oxide complex, not free nitric oxide. Since nitric oxide is, like oxygen, a heme diatomic ligand, NOS is subject to feedback inhibition by nitric oxide. Consequently, the competitive dynamics of nitric oxide-oxygen heme binding, which vary between the NOS and are determined by their varying rate of ferric heme reduction, ferric nitric oxide dissociation and ferrous nitric oxide oxidation, mean that INOS and NOS in particular partition catalysis into productive and futile cycles, yielding variable amounts of nitric oxide and nitrate-nitrite species. This is a delicate balance, which may all too easily be disturbed by immune inflammatory challenge and/or low cofactor availability, thus leading, yet again, to a lower nitric oxide peroxynitrite and related species ratio and pathology. L-arginine is bound at a distance to heme, its central N-guanidine 3.8 angstrom away from and coplanar to the heme. X-ray crystal data show that the dense network of hydrogens binding L-arginine to the NOS protein orients L-arginine and its N-hydroxy L-arginine intermediate rigidly in relation to the heme, with its hydroxylimin, oxygen, and guanidinium carbon distant from the heme ion by 4.3 angstrom and 4.4 angstrom, respectively. This distance is too long for an iron-oxygen bond, hydrogen bonded to the propionate group of the heme pyrrole ring D. Moreover, the substrate restricts oxygen binding. Thus, direct ligation to the heme is precluded, and N-hydroxy L-arginine is bound by NOS as the anti-stereoisomer to the supposed catalytic heme. There is also a distance of greater than 15 angstrom between the flavoprotein domain and the heme to which electrons must transfer. Moreover, the distance between the supposed catalytic heme's flavin mononucleotide module docking site for electron transfer or exchange between flavin mononucleotide and the cofactor tetrahydrobiopterin is even greater. Tetrahydrobiopterin being at least 17 angstrom away from the proposed flavin mononucleotide docking site, so that a through heme model for tetrahydrobiopterin radical reduction has been proposed, the electron equivalent of jumping through hoops. Therefore, a non-heme ion has also been proposed as the NOS catalyst, and it has been argued that tetrahydrobiopterin is too far away from the heme to directly participate in the chemistry of the reaction. Something is needed to bridge all these gaps. The enzymatically active form of vitamin B12, ADENOSYLCOBALAMIN, is known as cofactor for only one mammalian enzyme, mitochondrial methylmalonyl-CoA mutase, the enzyme which catalyzes the isomerization of methylmalonyl-CoA to succinyl-CoA. However, in microorganisms, ADENOSYLCOBALAMIN participates in a number of important enzymatic reactions, including those of ribonucleotide reductase, glutamate mutase, methylene glutarate mutase, dialdehydratase, glycerol dehydratase, ethanolamine ammonia lyase, and aminomutases. ADENOSYLCOBALAMIN's unique carbon-cobalt bond is potentially labile, and with the exception of ribonucleotide reductase in Lactobacillus leishmanii, ADENOSYLCOBALAMIN's mode of catalytic interaction within its dependent enzyme characteristically involves enzyme-induced homolysis of the cobalt-carbon bond with formation of a 5-deoxyadenosyle radical species. ADENOSYLCOBALAMIN-dependent enzymatic reactions fall into three classes. Class 1 involve reversible carbon skeleton rearrangements. Class 2 catalyze irreversible heteroatom eliminations. And Class 3 catalyze rearrangements with migration of an amino group. All have in common the 1,2 interchange of a hydrogen atom with a substituent group on adjacent carbon atoms of the substrate. If ADENOSYLCOBALAMIN takes part in NOS catalysis, it seems theoretically plausible that it may do so in a manner generally consistent with its characteristic chemistry in the known ADENOSYLCOBALAMIN-dependent enzymes. ADENOSYLCOBALAMIN is known to be produced at a rate in excess of the needs of mammalian mutase. It is not all protein-bound and is found in up to 4-fold higher concentrations than the methionine synthase coenzyme methylcobalamin in most mammalian cells. with the exception of fibroblasts where methylcobalamin predominates. Endothelial cells contain fourfold higher concentrations of adenosylcobalamin compared to cytosolic methylcobalamin in their mitochondria, where coincidentally mitochondrial NOS is also found. It has been observed in vitro that a significant proportion of exogenously administered cobalamin, after partial conversion to adenosylcobalamin, is found constantly associated with membranes in murine L1210 cells. Endothelial cells also express high levels of membrane-bound eNOS. Cobalamin supplementation of vegetarians with low cobalamin status significantly increases eNOS nitric oxide release in the brachial artery. Exogenous adenosine supplementation, which may increase adenosylcobalamin synthesis, increases synthesis of nitric oxide. Circulating leukocytes responsible for high iNOS nitric oxide in immune responses have the most extraordinarily high cobalamin concentrations, 38-fold higher than in erythrocytes. In mammals, adenosylcobalamin inhibits ribonucleotide reductase, but the mechanism by which it does this is unknown. Nitric oxide also inhibits ribonucleotide reductase, and during the cell growth cycle, the levels of both cobalamin and nitric oxide rise and fall together. Therefore, nitric oxide induction downregulates the multi-drug exporter 1 gene, MDR1, the cellular cobalamin exporter. MDR1 downregulation is also a known effect of cobalamin. It is notable too that both high nitric oxide and adenosylcobalamin cause cell cycle arrests at the G1 S phase boundary, the p53 cell cycle arrest checkpoint, whilst accumulation of nitric oxide leads to high expression of p53, which in turn inhibits iNOS nitric oxide post G1 S phase, with cobalamin levels falling in tandem. Such associations may not be coincidental. The accelerated rate of adenosylcobalamin synthesis with the generally high intracellular adenosylcobalamin-methylcobalamin ratio are both specifically dictated by the need to supply adenosylcobalamin, not just for the isomerization of methylmalonyl-CoA to succinate-CoA, but for regulated NOS catalysis and coupled nitric oxide synthesis. The adenosylcobalamin NOS-catalyzed nitric oxide then both directly by activation of DNA methyltransferase and SAM, the universal methyl donor, S-adenosyl methionine, with corollary nitric oxide regulatory effects on methionine synthase and methylcobalamin, the latter also known to act as a methyl donor, and indirectly via cell signaling networks regulates global methylation. Since there is a SAM methionine synthase cycle, cystathionine beta synthase glutathione cycle switch, that is determined by levels of homocysteine, the methionine synthase cystathionine beta synthase cycles have hitherto been viewed as intimately and critically interlinked, along with the methionine synthase-dependent folate cycle. It may be time, however, to pan out further and take a larger overview. The adenosylcobalamin-catalyzed NOS form a critical circuit together with the mutase, the TCA Krebs cycle, methionine synthase SAM cycle, the folate cycle, and the cystathionine beta synthase glutathione cycle, all continuously feeding into each other and ultimately all regulated, like all key eukaryotic biologic processes, by adenosylcobalamin-catalyzed nitric oxide. When the cofactor tetrahydrobiopterin is bound within each half of the NOS dimer, two molecules of free base-off adenosylcobalamin dock in the immediate vicinity, closer to tetrahydrobiopterin than the two thiolate-bonded hemes in the oxygenase domain, which they effectively block. The porphyrin rings of the hemes act as templates against which the corins of adenosylcobalamin correctly stack up in the active site. NOS adopt a closed conformation as each adenosylcobalamin dimethylbenzoimidazole simultaneously extends perpendicularly away from the active site, displacing the two upper zinc tetrathiolate cysteine bonds to form the zinc finger, ZNS2N2, which then functions as a nitric oxide responsive on-off switch for catalysis, as in matrix metalloproteinases. In the presence of the bound substrate L-arginine and tetrahydrobiopterin, homolytic cleavage of adenosylcobalamin's carbon-cobalt bond takes place, generating two paramagnetic centers, a 5-deoxy-5-adenosyl radical and cobalt in the cobalt-2 state. In a concerted fashion, cobalt-2 binds oxygen. As homolysis of the adenosyl radical's cobalt-carbon bond in adenosylcobalamin-dependent enzymes does not require an electron transfer, but is enzyme-activated, it seems unlikely that this will differ in the NOS. The tight fit of adenosylcobalamin's initially concave butterfly-shaped corin, coupled with the dense necklace of hydrogen bonds around tetrahydrobiopterin, which trap the adenosine moiety as they do in diol dehydratase, together with the substrate binding energy and perhaps some trans effect of the bulky extended dimethylbenzoimidazole base-off, should be sufficient to accelerate the homolytic scission of adenosylcobalamin by a factor of 10 to the power of -10 to 10 to the power of -12, a trillion-fold acceleration, typical of the effect on binding of adenosylcobalamin in most of its known dependent enzymes. Thus, at this point in adenosylcobalamin-mediated NOS catalysis, nicotinamide adenine dinucleotide phosphate-derived electron transfer from the NOS reductase domain is redundant. During the first monoxygenation, almost simultaneously with cleavage of the cobalt-carbon bond, the high-energy adenosyl radical abstracts a hydrogen from the terminal guanidino-amino group of L-arginine, which, together with one atom of dioxygen bound by cobalt in the 2 state, a thermodynamically privileged binding in 5 coordinate cobalamins by comparison with cobalt in the 3 state, and tetrahydrobiopterin yield the intermediate N-hydroxylated L-arginine as a substrate nitrosyl radical, adenosyl, oxidized dihydrobiopterin, the quinoid dihydrobiopterin, together with H2O and cobalt in the 2 state. The latter two products immediately combine as a conveniently highly unstable species, H2O CO2. This is the source of the single atom of oxygen required in the next step to convert N-hydroxylated L-arginine to citrulline and nitric oxide. The immediate proximity of a highly deprotonating agent, the zinc of ZNS2N2, to the active site may indeed also be intended to ensure rapid deprotonation for this purpose. In the second monoxygenation, adenosyl plus N-hydroxylated L-arginine plus dihydrobiopterin plus H2O CO2 initially yields citrulline and, since H2O CO2 or CO2 alone, though pertinently not adenosylcobalamin, can combine with nitric oxide, nitrosylcobalamin in the CO2 state, plus the adenosylcobalamin radical and 2 protons. Given nitric oxide's very high formation constant binding affinity for CO2, it seems likely that nitric oxide will, prior to its enzymatic release, react rapidly with CO2, transiently forming nitrosylcobalamin in its CO2 state, a highly reversible reaction found to be nearly diffusion-controlled. Within the NOS enzyme, this should prevent premature inhibition of NOS by build-up of nitric oxide. Further, If, for any reason, CO2 is oxidized during NOS catalysis to H2O-CO3 or hydroxo-CO3, which would also inactivate the enzyme, nitric oxide will immediately reduce it to CO2 and thence will bind it with transient formation of nitrosylcobalamin in the CO2 state. The possibility of this latter reaction has been established chemically on an electrode surface. Such a catalysis-related nitrosylcobalamin 2 transients within NOS might incidentally also explain the failure to detect endogenous nitrosylcobalamin intracellularly in vivo to date. To conclude, almost simultaneously to the formation of nitrosylcobalamin in the CO2 state, radical collision with the adenosyl radical displaces nitric oxide from nitrosylcobalamin in the CO2 state, thus avoiding the NOS futile cycle, enabling the geminate reformation of adenosylcobalamin. This brings the catalytic cycle to an end, with the subsequent enzymatic release of nitric oxide. The NOS reductase domain may be intended primarily to sustain the heme-based reserve reaction and thus redundant in the presence of adenosylcobalamin and tetrahydrobiopterin. That is, unless cobalt in the 2 state actually binds oxygen once more, rather than H2O for the second monoxygenation, in which case 2 nicotinamide adenine dinucleotide phosphate molecules would be required. In terms of cobalamin-dependent enzymes, there is some analogy between the NOS reductase as backup for the heme reaction when cobalamin is in short supply, and the role of the similarly flavin-based methionine synthase reductase, which chaperones methionine synthase and periodically reactivates it when the catalytic cobalt-1 is reduced to cobalt-2. However, given the relatively greater importance of NOS even than methionine synthase to sustaining mammalian life, nature takes no risks, which may be why eukaryotic NOS has a reductase domain inbuilt. Unlike most prokaryotic NOS, with the exception of Sorangium cellulosum NOS, which is phylogenetically related to eukaryotic NOS, prokaryotes also do not have a separate dedicated NOS reductase. But then most prokaryotes either synthesize or have ample access to adenosylcobalamin. Nature's choice of tetrahydrobiopterin as a catalytic cofactor is equally brilliant, for it is versatile enough to function either as a 1-electron donor in the heme-based backup reaction or as a 2-electron donor in the primary adenosylcobalamin-based reaction. In INOS, with rising high levels of nitric oxide, adenosylcobalamin dimethylbenzyimidazoles will ultimately be permanently displaced from their zinc anchor, as nitric oxide is known to displace trans-imidazole ligands, consistent with the known mechanism for NOS inactivation by nitric oxide. Moreover, since nitric oxide directly increases activity of DNA methyltransferase, it is likely that the INOS gene will ultimately also be hypermethylated or silenced. This hypothesis thus offers a fundamental mechanism for the complete biphasic regulation, early promotion and later inhibition of the NOS by increased levels of intracellular cobalamin, effected by nitric oxide responsive transcription factor SP1, promoting transcobalamin 2 upregulation during the normal inflammatory responses and immune challenge. Indeed, my colleagues and I have observed just such a biphasic regulation of the NOS, with promotion of initial levels of INOS-ENOS protein by high-dose cobalamins during the immune response to endotoxemia, with a corollary regulation of principal inflammatory mediators, interleukin-1, tumor necrosis factor alpha, COX-2, and high mobility group BOX1, and a consequent beneficial in vivo survival outcome. If this hypothesis is correct, the problem of designing NOS inhibitors, which has proven disastrous in sepsis, for malfunctioning NOS peroxynitrite-derived pathology is redundant. The clinical use of high-dose parenteral cobalamins should suffice to regulate the NOS and the nitric oxide peroxynitrite and species ratio. Beyond the persistent problems and mysteries of mammalian NOS catalysis, there are other indications that ADENOSYLCOBALAMIN may have an as yet unverified leading role in NOS catalysis. Nature is economic. Yet, in the mammalian cell, whilst ADENOSYLCOBALAMIN is found primarily in the mitochondria, it is found in excess of the needs of its known mammalian partner enzyme, methylmalonyl-CoA mutase. As long ago as 1995, Professors Ed Quadros and Don Jacobson also observed that the only known proteins likely to retain cobalamin in cells are the two enzymes methionine synthase and methylmalonyl-CoA mutase. The saturation state of these enzymes and total enzyme activity in liver and kidney can account for only a fraction of the cobalamin in liver and kidney. Therefore, a second look at cobalamin accumulation in these tissues is warranted. If nature is economic, what is the excess cobalamin, mostly ADENOSYLCOBALAMIN, in these organs and cells therefore? If ADENOSYLCOBALAMIN does take part in NOS catalysis as a third mammalian cobalamin coenzyme, it should be possible to detect this by studying activity of the whole NOS enzyme both in vitro and in vivo, comparing NOS protein levels and activity and nitric oxide synthesis in cobalamin-deprived environments, compared with the subsequent impact of ADENOSYLCOBALAMIN addition. It is noteworthy that a number of in vivo NOS studies have habitually or unwittingly expressed NOS in ADENOSYLCOBALAMIN-rich hosts. Thus, rat neuronal NOS, expressed in ADENOSYLCOBALAMIN-rich E. coli, shows a 4.5-fold greater turnover activity in the L-arginine to citrulline nitric oxide synthesis assay of 450 nanomole per minute per milligram of NOS protein, as compared to 107 nanomole per minute per milligram of NOS protein in purified rat neuronal NOS in vitro. Similarly, rat neuronal NOS from cerebellar extracts, transfected into human kidney 293 cells with significantly higher cobalamin than brain, demonstrate a 10-fold increase in NOS activity and protein over the cerebellar extracts. My colleagues and I have also seen a significant increase in NOS protein with various cobalamins in endotoxemic mice. Since administration of cobalamin is known to increase protein levels of methionine synthase, this may be another instance of the cobalamin cofactor increasing coenzyme assembly. It is also notable that studies of nitric oxide synthesis by prokaryotic NOS have only shown significantly high nitric oxide output when done in vivo, as in Streptomyces turgidis scabies, which contains ample ADENOSYLCOBALAMIN, and in Bacillus subtilis and Bacillus anthracis, expressed in E. coli. Since whole Bacillus subtilis in vivo in the human gut has an extraordinarily high uptake of cobalamin and an even slower ferrous nitric oxide dissociation rate in vitro than mammalian NOS, I would predict that an in vitro study of isolated Bacillus subtilis and Bacillus anthracis would, upon addition of ADENOSYLCOBALAMIN, show both higher nitric oxide synthesis and a high ferrous nitric oxide, or rather nitrosyl CoA2 dissociation rate when compared to isolated Bacillus subtilis controls without ADENOSYLCOBALAMIN. The same should be true of mammalian NOS. As we see, for example, rat neuronal NOS minus its reductase domain still produces nitric oxide, when expressed in adenosylcobalamin-rich E. coli. Other clues from bacterial NOS include the finding that a known adenosylcobalamin synthesizer, Lactobacillus fermentum, can also synthesize nitric oxide prior to the addition of tetrahydrobiopterin and flavin-reducing agents. The serendipitous finding by Bird et al. That NAD, nicotinamide adenine dinucleotide, added simply to improve crystallization for the elucidation of the structure of methicillin-resistant Staphylococcus aureus, bacterial NOS oxygenase, actually binds to the bacterial NOS as a ligand in the catalytic site, but has no catalytic effect and therefore results in no nitric oxide synthesis. This MRSA bacterial NOS can potentially accommodate a very large unknown ligand. Might it be that the molecular mimicry between the adenosine ribose of NAD and the adenosine of adenosylcobalamin points to an undiscovered large bulky molecule catalyst, namely adenosylcobalamin? Then there is the mystery of the general lack of a reductase domain in most microbial NOS, with very few notable exceptions such as some cyanobacterial NOS and the NOS of Serandrum cellulosum, which is phylogenetically related to eukaryotic NOS. Not only that, in spite of the absence of a reductase domain in bacterial NOS, there has been a complete failure to identify any dedicated redox partners. But if adenosylcobalamin is also the microbial NOS catalyst, then this failure and the redundancy of a reductase domain makes complete sense. Most prokaryotes either synthesize or usually have ample community access to adenosylcobalamin. However, NOS-containing eukaryotes are prone to cobalamin deficiency. Thus, the heme backup catalyst is essential for their pure survival. In adenosylcobalamin NOS enzymatic catalysis studies, electron paramagnetic resonance and UV-visible spectroscopy can be used to detect predicted accumulation of the CoA2 species in the NOS following L-arginine tetrahydrobiopterin adenosylcobalamin binding and adenosylcobalamin enzymatic homolysis. In all studies, electron spin resonance with spin traps should be used to detect true nitric oxide, and care should be taken to discriminate between nitric oxide and peroxynitrite-related species, including superoxide products of the NOS with and without adenosylcobalamin. The grease assay is not discriminatory. In pathologies associated with cobalamin deficiency, functional or otherwise, the nitric oxide, peroxynitrite and species ratio should be low, with high superoxide decreased or unproductive NOS nitric oxide activity and low levels of NOS protein. However, due to a phenomenon known as relaxed control of messenger RNA, when methyl groups cobalamin are in short supply, messenger RNA may paradoxically be high, a likely source of confusion, along with the grease assay, as to the real status of nitric oxide levels in pathology. Conversely, treatment with high-dose parenteral adenosylcobalamin or methylcobalamin, which will interconvert partially to adenosylcobalamin, should restore efficient NOS activity, a regulatory high nitric oxide peroxynitrite and related species ratio, and resolve inflammation. This wild imagining is offered as a possible new clue to the truth of still mysterious NOS catalysis, in the hope that, as Keats once wrote, the imagination may be compared to Adam's dream. He awoke and found it truth.